So we're going to finish up B cells, B lymphocytes as part of third line of defense today, and then we will move on to T cells, our T lymphocytes, and describe how they contribute as our specific third line of defense and how they do our cell-mediated immunity, right? So our B cells are doing what's called antibody or humoral immunity, because they are pumping out these antibodies, releasing them into the fluids, and those antibodies are the things that are doing, giving us our protection. Whereas our T cells, we said, give us cell-mediated immunity. So they're doing more of the hand-to-hand combat. So we're going to talk about how they do that, what they're effective against, what their weapons are, if you will, to kill off cells that they find shouldn't be there. Okay? So, I'm going to finish a few slides on B cells, then we'll do a review of what we were learning on B cells. So we'll look at like one of the crash course videos again, like we did last time, and then we'll move into new stuff with T cells. Okay? So before I do that, is there any questions in terms of procedural things or just general class things and questions? We're okay? Early on a Monday? Yeah? Okay. Let's jump into it then. So we had said that when we have an activated B cell that has identified its particular pathogen, we said that there was that whole method of what's called clonal selection, where we finally found the right B cell that has the right shaped receptor that's going to be effective against a particular pathogen at the time. That cell becomes activated and becomes one of two different types of cells. It either becomes a plasma cell, right, the big Hulk cell, the huge cell that starts cranking out those antibodies and releasing them into the fluids, like the lymphatic fluid as well as the plasma, or it becomes a memory B cell that will protect us, remember what we're infected with, and protect us in the future. So live to fight another day, right, rather than going into the battle right then. So when these plasma cells are producing antibodies, we need to be able to describe how these antibodies protect us. Now again, each time we go through any of these different lines of defense, we're thinking, okay, are these cells or molecules as part of first line, second line, third line, etc.? Obviously B cells, B lymphocytes are cells, but antibodies are just molecules, right? So these are proteins that are being cranked out and released that are floating around in our fluids. So how can they protect us? Well, one of the ways they can protect us we've learned about already, and that is through agglutination. So agglutination is kind of like I was mentioning, it's kind of like molecular handcuffs, right? So they have these little binding pockets to them, and if it matches up with an antigen, it will bind to it. And you can see how this particular antibody has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different binding pockets on it, right? So it can grab onto a bunch of these foreign cells with their foreign antigens on it, hold them all in one place, and create a clump, just like we saw when we were doing blood typing. And when we have a clump of cells, now they're not off randomly going around. We've located them all in one place, and now when we have a macrophage or a dendritic cell or something like that, they can come and ingest them and help to get rid of them very easily because they're all rooted in one place. So we've got agglutination. That's one example of how antibodies protect us. Neutralization is another one where neutralization, these things will often bind to smaller components, either like bacterial toxins that are being released or viruses that are not inside of cells that are just floating around on their own. They will come up and cover it, right? Like bubble wrap, so that it doesn't hurt our cells. So the viruses can't infect and get into our cells. So those toxins can't come into contact and affect our cells as well. So they surround it and bind to it and neutralize the threat. Okay? So neutralization is where it is masking or binding to, like we said, kind of toxins that are released by bacteria or even exposed viruses like that. Then also, there is precipitation. So precipitation, today there was a chance of precipitation outside, right? where there is rain, where we get this kind of condensation and it settles out of the sky. It comes out of, this more kind of nebulous thing and settles out. That's what's happening in precipitation as well, where you have soluble antigens that are floating around in the fluid, a little bit hard to access. If we start tagging them and binding these antibodies with them, they become heavier, if you will, and they kind of settle out of solution. Again, now it's easier for macrophages and things like that to find them, to ingest them, to get rid of them, as opposed to hunting them down in a soluble form. Yeah?
So is that sort of like sifting them out, kind of?
A bit, yeah.
Filtering them out a little bit?
Yeah, kind of like filtering. Like we said, it's a little bit like, when you're doing chemistry and there's a precipitant, right? So if you're swirling, and something starts to settle out just because it's too big or there's too much of it or something like that, you can see it in the bottom of the beaker. That's what we're kind of seeing here. So when it's in solution, you're like, I don't know, I don't see it, right? It's a sugar solution, it's salt or something like that. But when it settles out, you're like, oh, there it is. It's more obvious. And so now it's more obvious to our macrophages or phagocytes. Right. All right, so then we have precipitation. And then, in addition, antibodies, when they're tagging things, can help with other processes. So, for instance, antibodies can help to activate the complement system. So the complement system was part of our second line of defense. This is where we see this collaborative effort between third line and second line. Antibodies are produced by B cells. That's third line. But when they start tagging this foreign agent, they can work to activate that whole complement system, those series of proteins that will also start sticking to the surface of that foreign cell and form this membrane attack complex, this big hole in the plasma membrane that is going to cause the cell to basically burst or lyse. So they can start that process. They can act as a flag, if you will, helping to bring in those complement proteins and starting that membrane attack complex. So they can work with complement that can lead to lysing a cell, right, causing it to explode, killing off that agent. And then also they help in things like phagocytosis and inflammation. So for instance, in phagocytosis, they act as opsonins. So as an opsonin, when those antibodies are attached to that foreign agent, not only do they kind of make it more visible, right? It's like putting flags in that cell. Now it's much more obvious that that cell is something there that we need to pay attention to, that we need to get rid of. But in fact, as they're sticking off of that bacterium or whatever, they provide little handles, like little handlebars for the phagocytes to be able to use their pseudopods and grab onto to be able to more easily bring it in and ingest it and digest it and get rid of it. So it gives us a little bit more of that ability to latch onto it as well. So it enhances phagocytosis by acting as an opsonin, right? An extra little tag that gives it maybe a little bit of grip, if you want to consider it that, or a little bit of something to grab onto in order to make that process happen a little bit easier. And then inflammation as well. Again, just more tags that we have on these cells, and as these cells lyse and whatever, sometimes they can release other components. Histamine can be released as well as a B cell becomes activated, and that leads to inflammation, leads to more things arriving at that site of infection, etc. Okay, so the point here is what we're explaining is, as a third line of defense, we have to say what B cells do. B cells, in essence, in short order, produce antibodies. Okay, but for a complete answer for that, we have to say, okay, great, but how do antibodies help us to get over when we become infected with a bacterium or something like that? These are some of the examples of how antibodies protect us. Agglutination, precipitation, opsonization, neutralization, things like that. So one of the possible short answer questions is explaining the role of B cells. How do they become activated? Where do they come from? What do they do to protect us and stuff like that? So if you're going to explain how B cells work as third line defense, you've got to be able to explain that they produce antibodies and at least give some examples about how antibodies work. And that's what this is showing us. Okay? Okay. So then there are just a few follow-up slides discussing antibodies, right? So antibodies are also known as immunoglobulins. We saw that term a long time ago when we were introducing ourselves to the blood. We said there are different plasma proteins in there, right? Like fibrinogen and things like that, albumin, and then immunoglobulins. So this is a component of the blood, to be sure. And in fact, there are different types of antibodies based on their structure. The one that you saw with the blood, like that one has like 10 different binding pockets to it. They're not all that big. That one's pretty big. It looks like a snowflake, grabs onto a whole bunch of different pathogens, but they're not all that big. Some of them are just like you saw it, like you saw in some of the examples, like a simple Y shape where they have 2 binding pockets instead. So there are these different types of shapes, and these 5 classes of antibodies that you see there, the IG part stands for immunoglobulin, right? That's where that comes from. Then the D, M, G, I, G, A, and E are just 5 different classes, again, based on their shape, based on where they're found, and stuff like that. Don't stress about that. I'm not going to ask you, is this particular one an IgA, or is this an IgE, or whatever. But sometimes if you do get into immunology or blood typing or something like, not blood typing, but blood analysis, they may look at, okay, what types of antibodies are elevated, trying to give them a little bit more information about, kind of what you're infected with, where it is, and things like that. So this is a kind of generic layout of one of the simpler antibodies. And what you can see is this area of it here. So this area here, this heavy chain region, this is where, like as a receptor, it was originally bound to the B cell. Because remember, B cells, all the different B cells have these receptors sitting on their cell surface that allow them to identify a particular agent. And then once we figure out which B cell we need, based on these areas, right? These areas here are where, these are the binding pockets for that foreign antigen. Based on the shape of those, then we start cranking out these antibodies. We release these antibodies, which have the exact same structure as the original receptor. But we start releasing them from the cell, and then this whole thing is what's bumbling around, right? So those are areas, like we said, that would bind to the foreign antigens if they came into contact with them. These areas are where macrophages can come up and grab onto them, right? We said it can work as an opsonin, helping give it some grip. So it has little binding sites for the macrophages to be able to grab onto it as well. So there are specific aspects to the protein molecule that are important. Places where it binds to the foreign antigen, original places where it was attached to the B cell, if it was a receptor and things like that. But we're not going to get into that. I'm not going to ask you about the light chain variable, constant region, things like that. The only thing that I'll point out is where it says variable region, that's where we have that variation in terms of the different shaped receptors, right? There are millions of different types of B cells based on the little binding pocket shape. And so those variable regions that we see right there is where it'll be slightly different between B cells, and that way it can potentially match with a foreign agent that we get infected with in the future. Okay, so that is wrapping up our B cells. So what I'd like to do right now is pop over to watch a little bit of a review using our crash course instead. Because I think they do a good job of kind of bringing it home.
Time to hit refresh. New shoes just dropped at DSW.
What's true in World of Warcraft is also true in your immune system. To defeat your enemy, you have to know your enemy. Uncover its weaknesses. Learn how to see it before it sees you. We've already talked about how your innate defense system keeps out or quietly neutralizes pathogens without too much fuss. But sooner or later, a threat's gonna come along that's stronger than what the first responders can handle. That's when it's time for the adaptive or acquired immune system to step in. While your innate system takes its zero-tolerance policy very seriously and tries to toast any foreign microbe that it encounters, your adaptive system does things differently. It has to be expressly introduced to a specific pathogen and recognize it as a threat before it will attack. As its name suggests, you're not born with a working adaptive immune system. It's slow to act, in part because it takes time for it to shake hands with so many pathogens and get to know them. These introductions may be organic, like touching a dirty faucet in the bathroom or walking into a sneeze cloud, or they may be premeditated, which is why vaccination is pretty much the greatest thing to happen to medicine ever. But once it's been introduced to a potential threat, your adaptive defense defenses never forget it. And this ability to remember specific pathogens is one of the key differences between the adaptive and innate defenses. Another main difference is that adaptive immunity is systemic. Rather than being restricted to a particular infection in, say, a sinus or a sliced finger, your adaptive system can fight throughout your whole body at once. And it does this by deploying one or both of its separate but cooperating defenses, your humoral immunity and your cellular defenses. Your humoral immunity, which you might not have heard of before, works by dispatching important proteins that I'm sure you have heard of, antibodies. They're made by special white blood cells, and they patrol the body's humors, or fluids like blood and lymph, where they combat viruses and bacteria moving around the interstitial space between your cells. Much of what you know or have heard about or think of when your immune system comes up actually has to do with your humoral immunity. It's why if you had mumps as a kid, you probably don't have to worry about getting it again for the rest of your life. It's also why doctors and nurses and patients who have been infected with the Ebola virus, a disease once thought to be incurable, have lived to tell about it. And it's why vaccinations work. Whether you're protecting yourself from infections or playing an MMO, one of the first steps in any good defensive strategy is to be able to tell your friend from your foe. And in the case of your immune system, that means being able to identify antigens. An antigen could be an invader from the outside world, like a bacterium, virus, or fungus, or it could be a toxin or a diseased cell within your own body. But in any case, antigens are large signaling molecules not normally found in the body, and they act as flags that get the adaptive immune system riled up. So let's say flu virus gets inside of you, and it's floating around trying to find a good host cell to start multiplying inside of. Before it finds that cell, hopefully it will be paid a visit by one of the stars of your humoral response, a B lymphocyte. Like all blood cells, these guys originate in your bone marrow, but unlike other white blood cells, they also mature in the bone marrow too. And as a B cell matures, it develops the ability to determine friend from foe, developing both immunocompetence, or how to recognize and bind to a particular antigen, as well as self-tolerance, or knowing how to not attack your body's own cells. Once it's fully mature, a B lymphocyte displays at least 10,000 special protein receptors on its surface. These are its membrane-bound antibodies. All B lymphocytes have them, but the cool thing is every individual lymphocyte has its own unique antibodies, each of which is ready to identify and bind to a particular kind of antigen. That means that with all of your B lymphocytes together, it's like having 2 billion keys on your immune system's keychain, each of which can only open one door. So part of your immune system's strategy is just to win with overwhelming odds. The more unique antibodies your lymphocytes have, the more likely it is that one will eventually find, bind to, and mark a particular antigen. Once they've matured, B cells colonize or seed your secondary lymphoid organs like your lymph nodes and start roaming around in your blood and lymph. At this point, they're still naive and untested, and they won't be truly activated until they meet their perfect enemy match. Which brings us back to the flu virus. When the right B cell finally bumps into an antigen it has antibodies for, usually in a lymph node or in the spleen, and recognizes it, it binds to it. This summons the full power of the humoral immune response, and the cell basically goes into berserker mode. Once activated, the B cell starts cloning itself like crazy, quickly producing an army of similar cells, all with the instructions for the exact same antibodies that are designed to fight that one particular antigen. Most of these clones become active fighters or effector cells, but a few become long-lived memory cells that preserve the genetic code for that specific successful antibody. This ensures that if and when the antigen returns, there will be a prepared secondary immune response that's both stronger and faster than the first. This is key to why vaccinations are so brilliant and important, which I'll come back to in a minute. But while the memory cells are just there to hang back and record things, the effector, or plasma cells, are packed with extra amounts of rough endoplasmic reticulum, which acts as an antibody factory. These cells can mass produce the same antibodies over and over for that particular invader, spitting them out into the humor at a rate of around 2,000 antibodies per second for 4 or 5 days until they die. And the antibodies they make work the same way that the membrane-bound ones do. They're just free-floating. So they ride the tides of blood and lymph, binding to all the antigens they can find and marking them for death. Now, antibodies can't really do the killing themselves, but they do have a few moves that could make it hard for intruders to take hold. One of their most effective and common strategies is neutralization, where antibodies physically block the binding sites on viruses or bacterial toxins so they can't hook up to your tissues. And because antibodies have more than one binding site, they can bind to multiple antigens at the same time in a process called agglutination. The resulting clumps can't get around easily, which makes it easier for macrophages to come and gobble them up. And not only that, but while all this is going on, antibodies are also ringing a chemical dinner bell, calling in phagocytes from the innate immune system and special lymphocytes from the adaptive system to destroy these messy little antigen antibody clumps. So the point of all of this in the short term is to keep you healthy, but in the long term, this process also adds to your overall immunity. The humoral response allows your body to achieve immunity by encountering pathogens either randomly or on purpose. Active humoral immunity is what we were just talking about. It's when B cells bump into antigens and start cranking out antibodies. This can occur naturally, like when you catch the flu or get chicken pox or pick up some nasty bacterial infection, or it can happen artificially, particularly through vaccination. Most vaccines are made of a dead or extremely weakened pathogen, and they work on the premise that because a secondary immune response is more intense than a primary response, by introducing a pathogen into your body, you're priming it to fight hard and fast should that antigen show up again. In the case of typically non-fatal infections like the common flu, this immunity should at least spare you from some of the most severe symptoms. But in the case of more serious diseases like polio, smallpox, measles, and whooping cough, vaccinations can be truly life-saving. Now, some antigens, like those from mumps or measles, don't really change much over time, so a few immunizations will leave you set for life. But others, like influenza, are constantly evolving and changing their surface antigens, so immunity to last year's flu probably doesn't work against this year's flu. Still, acquired immunity doesn't have to be active. Babies, for example, naturally obtain passive humoral immunity while still in the womb. They receive ready-made antibodies from their mothers through the placenta and later on through breast milk. And that works pretty well for a few months, but the protection is temporary because passively obtained antibodies don't live long in their new body, and they can't produce effector cells or memory cells, so a baby's own system won't remember an antigen if it gets infected again. You can also acquire this kind of temporary passive immunity artificially by receiving exogenous antibodies from the plasma of an immune donor. This is what recently saved some doctors and nurses who had contracted the Ebola virus from infected patients. A serum was made from the blood plasma of other medical workers who had been infected and survived. The antibodies helped defend the patients from the virus before their own active immunity could identify that particular antigen and start creating their own antibodies. It's not the same as a vaccine, which immediately engages your B cells, but it can buy a patient some crucial, life-saving time against an infection that would otherwise quickly kill. But B cells and antibodies are only part of the immunity equation. There are plenty of pathogens that quickly worm their way right inside your cells, where they're safer from the humoral response and free to multiply as much as they'd like. Luckily, your immune system has yet another game plan and a new set of players ready to fight that final battle with cell-to-cell combat. Make sure you catch our final episode next week and learn all about this epic battle royale. But as for today, in our second to last episode, you learned how the adaptive immune system's humoral response guards your extracellular terrain against pathogens. We looked at how B cells mature, identify antigens, and make antibodies, and how antibodies swarm pathogens and mark them for death. We also talked about active and passive humoral immunity and how vaccines work. Thank you to our headmaster of learning, Linnea Boyev, and thank you.
So that was good, especially towards the end there as well, because do remember that we talked about that as a topic too, where we were discussing active and passive immunity, then naturally acquired, and then artificially acquired. And that, as you're looking through your exam objectives, is another one that's in bold. So a possible short answer question where you kind of have to compare those different terms. Okay? Okay. So before we then move on to T cells, the final part and final player of our immune system, questions of anything to date. First line defense, second line defense, B cells, etc. Dead silence is a no. Okay. Don't forget, on Mondays, I have office hours lots of days, but on Mondays from 1 to 4, I'm in the study lab as well. So not only for lab things, but also for lecture things if you want to go through and ask questions or just kind of go through things and review things. Okay, let's pull into T-Town. So this is talking about third line of defense, the T lymphocytes. So again, what we're going to see here is very specific, have memory cells. And again, this is one of the things to think about too. So each of our lines of defense, we're thinking, okay, so what are my examples of cells and molecules? So even when we were doing third line of defense with B cells, we kind of had two different types of B cells, right? We had the plasma cells, the activated B cells that go into the fight, and then we had memory B cells. Those are two different types of B cells that you could discuss, that you could use as examples for third line of defense. with T cells, we even have more. So for our T cells, and we'll talk about a few of these, we've got helper T cells, we've got regulatory T cells, and we've got cytotoxic T cells. And for all of those, we also have memory T cells. Just like in our B cells, we have memory T cells that are going to give us that long-term protection, okay? So they have been activated, but they will protect us and recognize if we get involved with that foreign agent in the future. All right, now, in terms of our review of T cells, we're going to focus mainly on helper T cells and cytotoxic T cells. As you can see in terms of regulatory T cells, it is another T cell type. These ones help to moderate the response of other T cells, making sure that we don't overreact, right? We talked about like allergies and things like that, which are kind of overreactions. But there are also times when, as we're creating this clone army, we may kind of go unto excess, and we don't want that to happen. And so regulatory T cells are helping to moderate that response, so we don't over-respond. And so we won't really talk about them in too much more detail, but if you were looking for another example of a T cell and, describing what it does, you could use regulatory T cells as well. All right. So what we're going to do here is we are going to look at these different types of T cells. We said there are helper T cells, and we said that there are cytotoxic T cells. And their common names give us an idea about what they do, right? Helper T cells in some way are going to help. A little bit kind of like antibodies, right? You're like, well, they're going to help. Maybe they're not going to fight off the invader themselves, but they're going to help in some way. So we're going to take a look at that. And then cytotoxic T cells, that name, right? Cyto means cell, toxic basically means poison. Oh, so those definitely are going to be those T cells that are going to be doing our hand-to-hand combat. The other thing that I'll have you know, so here we can see our different T cell lines, kind of where they come from, etc. But the other thing that we can see here is take a look at this. We're seeing examples of our MHC proteins coming into play again. And if you'll remember, an MHC protein is a major histocompatibility complex. That's what it stands for. An MHC protein is a self-antigen. It tags our own cells, for the most part, to say, oh, this is part of our own body, let's not attack it, right? Let's protect it, okay? But we did mention that Class 1 MHC proteins on a normal healthy cell, whether it's a skin cell or a muscle cell or something like that, if it's a healthy cell, it basically says, hey, I'm on your team. Look, here's my pass. I'm on your team. Don't attack me, right? But we do know, like we saw in some of the videos, sometimes we have intracellular pathogens, like viruses that can get inside of our cells. Now that cell, which is of our own body, is infected, it's diseased, etc., so we need to kill it off. So we need to have things like our cytotoxic T cells be able to very closely look at those MHC1 molecules to make sure it's healthy, that cell is healthy. And if it's not, it'll actually show up, that MHC1 molecule will be kind of corrupted, and that will set off a T cell immune response. It'll recognize that, okay? And that's why T cells are more effective with intracellular pathogens, right? Those antibodies are just bumbling around in fluids. They're not going to be able to very accurately say, well, you are one of us, but you don't quite look right, right? A T cell can do that. They can very closely inspect our own cells to check out to see if they're diseased, if they're cancerous or something like that, right? Because that's a trickier situation. It's our own cell that has self-antigens on it, but it's become diseased. Okay, we said class 2 MHC proteins are, again, it's a self-antigen, but those are only found on our antigen-presenting cells. So that APC that we see there stands for antigen-presenting cells. So antigen-presenting cells have both MHC1, because they mark it as part of us, but they also have MHC2. And that's going to be key in terms of, like we said, showing it off. I've eaten something that was really disgusting. I want to show you what it is because we're infected with it. So other people, other cells should know, can you help me with this? This is in here somewhere with us. Be on the lookout for this. Go out and check and hunt for this. Okay? Like a wanted poster. So the MHC2 molecule is only found on antigen-presenting cells, and it is used to present those foreign ingested organisms with their foreign antigens, right? So we'll talk about that a little bit more as well. So now we're getting into these T cells that are able to inspect these different types of self-antigen and process them and then respond as necessary, okay? So T cells are very effective against things like cells that are infected with viruses. There are some intracellular bacteria, some very small ones, and other types of within cell parasites, stuff that's hiding inside of our cells. It's also good, T cells are also good for cancerous cells, our own cells that are now replicating out of control, right? We got to be able to recognize that. It's us, so to a certain extent we're conflicted. We should protect it, but it's us and not functioning right. So we need to get rid of it, okay? And then also, if we have had some sort of a transplant or something like that, right? If you were receiving blood in the hospital and it was the wrong type of blood, we need to kind of be able to recognize it. So whether it's blood or other types of foreign tissue or something like that, we need to be able to inspect it and, form an alert as necessary. Okay, so like we said, we're seeing these T cells that as a living cell can more closely inspect our cells or cells in our body and look to see about if they include self-antigen, like the MHC molecules, and then do they have any aspect of a non-self or foreign antigen in them as well. Whether it is an infected cell, which is kind of showing some of that foreign antigen with that MHC1 molecule, or whether it's an antigen-presenting cell, which is doing it on purpose and saying, hey, I ate this, let me show it to you. Don't attack me, right? Here's my pass, here's my self-antigen, but as part of it, here's the foreign antigen. This is the thing I want to show you, okay? So T cells are going to be recognizing those combinations of foreign antigen and self-antigen, okay? This slide just goes through those two different types of MHC molecules. And I think MHC molecule is like a vocabulary term or something like that. So you need to know it's a major histocompatibility complex, right? It tags our cells as compatible with our tissues. Okay, so let's take a look then and see about our different types of T cells. We know that they're inspecting cells. We know that they're looking at these self-antigens, but they're also looking to see is there any foreign antigen in there as well, okay? Okay, so we said we're focusing mainly on helper T cells, cytotoxic T cells, and then of both types, we do have memory T cells too. So again, if you need another type of example of a T cell, a specific type of T cell, you can just say, oh, well, there are memory T cells that will protect us from that same pathogen in the future at a later date. All right, so let's look into the detail then of our main cytotoxic T cell, okay? So this is going to be our type of very specific killer cell, if you will. They are related to natural killer cells, but they're very, very specific. So what we see here is our inactive cytotoxic T cell, right? Going around in our lymphatic tissue or our fluids or something like that. They're coming up to an infected body cell, right? One of our own cells. Then what they have on the cytotoxic T cell, what they have is what's called a CD8 protein. And this is why some of these cytotoxic T cells are sometimes called CD8 cells, because they have the CD8 protein. And notice it has two golf club parts to it. This one is kind of a 4, and this one's a 4. So if we have two 4s together, that's 8, right? And that's kind of where the name comes from. So it has this receptor called the CD8 receptor, and what that CD8 receptor does is, notice it comes out and it inspects and binds with, like a docking marker, it is going to inspect the MHC1 molecule. So the first thing we check is, do you have an MHC1 molecule? Are you on part of our team? If you're not on our team, oh, there's all kinds of other things, phagocytes and natural killer cells that are going to kill you anyway because you don't belong here at all. But the first thing we check are you part of us? And so the CD8 molecule checks the MHC1 molecule that you can see there in green. Okay? That's the first thing. Okay, you're on our team. Now we want to see, are you a healthy cell and we leave you alone and protect you? Or are you infected with something? Is there something inside of you? Like if you are more like a zombie or something like that, we need to take a closer look at you and make sure that you're Uncle Joe, yes, but oh, are you Uncle Joe that's been infected with the zombie virus and now you're a zombie? And even though we're related at one point, we need to, right, chop your head off or whatever, okay? So there is a second receptor that you see right here. The second T cell receptor right here is inspecting the MHC1 molecule to see if there is any foreign antigen in there as well. So first we check the MHC1 molecule with the CD8. We say, oh, okay, you're on our team. Now we want to see if you're healthy. We're going to check this other binding pocket and see if there's a foreign antigen that's in there as well. Okay? If there's not, good. You're healthy, you're clean, we're moving on. If there is, now we need to generate an immune response. We have found some intracellular pathogen that's hiding inside that cell that we're infected with. We need to raise the alarm. So when we raise the alarm, third line of defense, what do? We create that clone army. We need a whole bunch of that particular T cell, which has the correct shaped receptor to bind with that particular foreign antigen, right? That foreign antigen is round. We have a round shaped receptor. It fits with it. That's the T cell that we need. That's the T cell that is going to be effective against any other cell in the body that's infected with that same pathogen. So we need a whole clone army, like in Star Wars, that whole clone army, right? We need a whole clone army of that particular T cell. So we've activated that T cell. It becomes a whole clone army of cytotoxic T cells that are going to start inspecting all kinds of different cells of our body to see if other cells are infected with that same virus or whatever it is, okay? In addition to that, of course, we're going to be making memory cytotoxic T cells that'll be long-lived, that are not going to go in the fight, that will be in our system on patrol for if we get infected with that again. Okay? Okay. So what we're seeing here is a cytotoxic T cell inspecting our cells, looking at that MHC1 molecule, and seeing if it has a foreign antigen associated with it. Okay? If it does, it's going to spark an immune response. If it doesn't, okay, you're clear, you're healthy, let's move on. Okay? Okay. So, as we were saying, cytotoxic T cells are going to be good at inspecting our own body cells, and their targets are going to be things like virus-infected cells, cells with other intracellular pathogens, cancerous cells, and then other types of foreign cells from like transfusions or transplants. So once those get activated, then we'll have to finish out that story and say, okay, we have this whole army that are going out into the war. What are their weapons that they're going to use in order to kill off infected cells that they find? So we'll see that coming up. All right, but before we do that, what I want to contrast with is I want to contrast that with our helper T cells. So our helper T cells are doing a similar thing. They're looking at cells of our own body, but helper T cells are only interacting with antigen-presenting cells, like macrophages, dendritic cells, and things like that. So again, they are also having a foreign antigen presented to them. This is a big macrophage. Oh, I ate this. I'm going to show it off, right? So you see, here is an inactive helper T cell that's arriving at an antigen-presenting cell, a big macrophage. And look, here we go again. There's a CD4 protein. So we have only one CD4 protein, only one of these golf club-like receptors. It goes out and it double-checks the MHC2 molecule. So it says, oh, okay, are you on my team? Okay, you're on my team. You're okay. I'm not going to attack you, right? Because I can see you have the MHC, our self-antigen. But then this other T cell receptor here is going to be checking for what you're trying to show me. Where's that foreign antigen? What do you want to show me? And just like we saw with the cytotoxic T cells, if we have the right helper T cell that has the complementary shape for that particular foreign antigen, then we're going to need a whole clone army of that helper T cell. And we're going to produce a clone army of that helper T cell. We're going to produce another smaller contingent of memory helper T cells against that foreign agent as well that we'll keep in the circulation. Okay? So the activation process is very similar. It uses instead like 1/2 a CD4 protein instead of the double CD8 like we saw in the cytotoxic, but it's doing the same thing. We check the self-antigen first, make sure you're on our team, then we use that other T cell receptor and see if you have a foreign antigen, and if you do, does it fit my receptor? If it doesn't, let's try another one. Does it fit this receptor? Nope, move that cell out of the way. Does it fit this one? So it takes a bit of time to figure out which clone we're going to need to be effective against that particular pathogen that we're being shown. Okay. So now we have the right helper T cell with the right shaped receptor. We produce this clone army. How is that clone army going to help? How, right? That's the name of the cell. It's a helper T cell. How is that helper T cell, that army, going to help us? Well, in fact, these helper T cells, what they do, and in fact, it's as simple as, I may have it on another slide as well, but their function is as simple as what you see down here. So helper T cells secrete cytokines. These are intercellular messages. And this IL stands for interleukin-2. So inter means between. Leukin reminds us of leukocytes. That stimulates white blood cells. So what this is doing is it, as a helper T cell, is releasing chemicals that amplify the response of other cells. Most notably, interestingly enough, helper T cells are releasing these chemicals that will stimulate the B cells to replicate more. So a B cell that's also become activated that needs to make a clone army, we get these helper T cells that also recognize that. They're like, hey, I can help with that. And they start releasing these cytokines or interleukins that then increase the speed at which that B cell army is created, right? So with the B cells creating this army to release antibodies, if we don't have the helper T cells cheering it on with these cytokines, we get a much, much weaker B cell response and a much, much smaller amount of antibodies produced. But if you do have these helper T cells that are releasing these cytokines, it will produce a massive B cell army that will then release a whole bunch of those antibodies that we saw in the video to then start agglutinating and neutralizing and stuff like that, okay? So while these helper T cells, you're like, oh, okay, so they're like cheerleader cells. So that's nice, but is that really helpful? It's definitely helpful. Like I said, if you don't have these helper T cells, your B cell response, your ability to produce antibodies is way, way weaker, okay? So it's really important that we have these helper T cells releasing these stimulatory chemicals for the B cells to then produce that massive B cell response, okay? So sometimes you see them designated this T sub H, that's the helper T cells like that, right? So it can stimulate a lot of our B cells like we talked about. It can even stimulate other types of T cells. So the proliferation, the creation of our lymphocyte armies are really amplified by helper T cells. So they actually have a really important role. And interestingly enough, and this will come up later on, when we talk about HIV, which if you haven't talked about HIV before, you should, as well as other types of, disease-causing agents, maybe sexually transmitted. But that one's called a human immunodeficiency virus, right? We're talking about the immune system. If you're deficient, that means it's compromised. It's not working well. This is a virus that selectively attacks helper T cells. So the HIV virus, HIV, the human immunodeficiency virus, infects helper T cells. So if you knock out helper T cells, now what happens is your other T cells, your cytotoxic T cells, and your B cell responses are much weaker. So most people that die of AIDS, right, the immunodeficiency syndrome, don't die of AIDS itself. They die of other infections that they can no longer defend themselves against because their immune system is compromised. because those helper T cells are knocked out. So their normal T cell and B cell response is much weakened. So they'll die from, things like whatever. It could be a flu virus or pneumonia or something like that. They just can't fight things off. That's why they have to be secluded and things like that, because their immune system is compromised by this virus, which is attacking their helper T cells. Okay. So helper T cells are getting activated by our antigen-presenting cells. So that's the relationship. Our antigen-presenting cells are showing off to our helper T cells. When they become activated, their way that they help us is by releasing these cytokines and interleukins that are like cheerleading chemicals that then amplify the response of our cytotoxic T cells and our B cells. Okay. So what we need to wrap around to then is we describe how our cytotoxic T cells become activated, but the question is now, what are their weapons, right? They've been able to identify some sort of intracellular pathogen, but now we need to find out what kind of weapons do they have. Once they've identified some sort of disease-causing agent, how can they kill it off, right? And so the way that they do that is that these are their kind of different weapons we see listed here. So there are things that are called granzymes. So granzymes, as you can imagine from that suffix there, these are enzymes that trigger apoptosis. If you remember that term, this is where we're telling a cell to die off, right? So it triggers this programmed cell death, which is really important because we already have a cell of our own body that's infected with a virus. It's a goner. The only thing it's going to be doing is replicating more virus and releasing that and infecting other cells. So we need to kill it off, right? So granzymes help to tell that cell, it's your time to die, give it up, okay? We also have things like perforins being released. So as that name indicates, they create perforations. They poke holes in the plasma membrane of that cell, again, causing it to lyse. There are also other things called granulysins as well as lymphotoxins. These are also other enzymes that if we poke these holes into that infected cell, now these kind of toxic chemicals, right, because it's a cytotoxic T cell, these toxic chemicals can get inside of the cell and help to degrade and kind of eat it from the inside out, okay? So that's kind of what we're showing in this figure here. So here we see a T cell, a cytotoxic T cell that has found an infected body cell. So it's looked with its CD8 receptor, it's identified the MHC1 molecule, but it's also used that other receptor to find that it's infected with some sort of foreign agent, right? So it has marked this particular body cell for death, and you can see it has these little vesicles in here that are filled with its weapons. And so it docks with it, if you will, and starts raining down these chemicals onto the surface of it. And these little molecules are the perforins. And you can see how, kind of like the complement system is not specific, but it works very much the same. A bunch of proteins go doop, 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 and they start creating this membrane attack complex, a big pore or a hole, and we've now poked a hole in the cell. So we do that here with perforins. But these perforins not only cause lysis, but they allow these other chemicals, like these other digestive enzymes, like granzymes, to get into this cell and cause it to die off, cause it to lyse, blow up, if you will, also cause it to die off. Okay? They kind of eat away at it on the inside. So in a cytotoxic T cell, these are some of the molecules associated with a cytotoxic T cell that are its weapons. Things like granzymes and perforins. And so you need to be able to kind of list those. What's a granzyme? What's a perforin? What's the cells associated with it? And then describe what it does. Okay? And like usual, you don't have to list every single thing that it does, as long as you have some examples of how a cytotoxic T cell works, or a helper T cell works, or a B cell works, etc. Okay? Okay. So, what we're seeing here, right, in terms of the cytotoxic T cells and natural killer cells are cousins, if you will. And they produce what's called immunosurveillance, which is probably another vocabulary term, if I remember correctly. So immunosurveillance, right? Surveillance is where something is being watched closely. In this case, our own cells are being watched closely. We're inspecting them all the time and checking them out to make sure that they haven't become cancerous, that they haven't become infected with a virus or something like that. And if they have, we're going to kill it off. Now, the natural killer cell doesn't have memory, not going to be able to identify specifically what virus it is, but it is able to inspect cells and help to kill it off. The cytotoxic T cells are important to be able to protect us long term. So we mount a whole clone army against that particular pathogen, and we produce memory cells so that we're protected against it in the future as well, right? It's the whole idea, if we think back to some relevant examples in current times, I don't know, COVID, right? So COVID is a virus that gets inside of our cells. So it infects inside of our cells. It starts with like respiratory cells and things like that, right? But we have to be able to identify our infected cells and then get rid of them. And we want to be protected in the future. So if you've had COVID, or if you've gotten the COVID vaccine, you should have some type of protection, right? Now the problem is, like we were seeing in the video, is that a COVID virus, like some other types of viruses, change and mutate over time pretty quickly. And so that's why you hear about variants. Like the very first one, the ones that are going around now, which are not as common because, again, people have this memory, because they have memory cells in their body, that if they're getting infected with it, they're not getting sick and they're not spreading it anymore, so it's not as much of a problem. But those original COVID viruses are quite different in terms of their foreign antigens than the ones that exist now. And so we have to have, that's why there are updated vaccines and things like that. And that's why you can still potentially get sick with COVID. You're like, I already had it once, or I had the vaccine. Why am I getting sick? Two reasons. Number one, because you may have a new variant that your immune system only partially recognizes or doesn't recognize at all. And so we're having to do this whole process of, okay, which is going to be the right T cell that's going to be able to help us out with this and things like that. We have to go through that whole process again. Or it's different enough that it takes us a little while to recognize it, right? It has almost like a different mask to it. You're like, hey, you look familiar. Got to take some time to figure out, have we been exposed to this before, have we not, etc. Okay, so cytotoxic T cells do give us that long-term defense because they do produce memory cells. Natural killer cells are related to them, and they use some of the same weapons in terms of perforins and granzymes and stuff like that, but they don't give us their memory cells. But collectively, natural killer cells, which are part of our second line of defense, non-specific, and our cytotoxic T cells give us that immunosurveillance, where we're inspecting our tissues and our cells all the time to make sure we're not infected with something. Okay. Maybe. We'll see if it's here. Do we have it here? Is it embedded? It is. Okay. Sometimes, again, with cells and molecules, it's a little bit
This patient's
easier.
cold is caused by
You can
rhinoviruses,
see animation.
shown here in red, that entered her nasal passages and escaped the first-line innate defenses, such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells, where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out
Dendrites
surveillance
sticking
for
off,
anything
right?
that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus.
So we're killing it off.
The resulting rhinovirus fragments are called epitopes.
So epitopes are part
The
of those
dendritic
foreign
cell
antigens
then
that we're
attaches the rhinovirus epitopes to presentation molecules called MHC2,
killing. So parts
This
south,
complex
foreign,
folds
shown on the
outward.
surface.
so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node.
It looks like an alien.
Here, the
but that's part of
dendritic
your
cell,
body
part
going
of the
around,
innate
right?
defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2.
So we don't attack
The
that
dendritic
cell,
cell
right? We gotta
then
verify
stimulates
that it's part of
activation
us as
of
well.
this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus,
So again, epitope is basically...
but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2.
So that's the interesting thing.
Our
B
mature
cell can
helper
actually
T
be an
cell
antigen-presenting
recognizes
molecule
the
as well.
rhinovirus epitope with its TCR and the MHC2 with its CD4. Then the helper T cell secretes chemical messengers known as cytokines
So those are
that
the cheerleader
activate the B
molecules.
cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell.
So what we find
With the help
is
of
that
cytokines from
those
a nearby
antigen-presenting
helper
cells
T
can
cell,
show off,
the
and it's
cytotoxic T cell
uncoordinated.
is activated and clonal
Like
expansion
disease cells
occurs,
show off.
resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1.
So there it is again.
The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Together, the humoral and cellular defenses are the body's third line of defense in fighting a very specific pathogen. The humoral defenses, ultimately in the form of antibodies, conduct surveillance for rhinoviruses that are outside the body cells, in the patient's tissue spaces and bloodstream. The cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory T and B cells help our patient's body to recognize and more rapidly fight the same rhinovirus should it infect her again.
So a key thing about that, like we were seeing, is that we get that memory from it. We see our T cells and our B cells becoming activated, but we see that the B cells are producing antibodies, and the antibodies are what are going on and binding with these foreign agents, whereas the T cells are going on and inspecting cells. So they're inspecting whether it's an antigen-presenting cell or whether it's an infected body cell. They're inspecting these cells and looking for these intracellular pathogens, right? So the antibodies that are produced by B cells are more effective against like bacteria or extracellular pathogens, or their toxins or waste products and things like that. So if it's hiding inside of a cell, then that's going to be more the job of our cytotoxic T cells instead. Okay, so this is what I was talking about. Oh, I'm sorry. Yeah, go ahead.
About that video,
Yeah.
can you simplify it down to the statement, or maybe I'm missing something? The MHC2 is to show it off, say, here, look at what I've got, and then the cell, depending on what type it is, will reproduce as necessary. And then the MHC1 is showing off that I'm infected, please address this by apoptosis. Is MHC1 always showing off that the cell's infected, or is it more complicated than that?
In a general way, that's correct. The only minor aspect to that, and that we did see in the video, is that an antigen-presenting cell, which is one of our body cells, but its job is to ingest these foreign agents, it can show off both of them. It can use the MHC2 to show off specifically to our helper T cells and say, hey, which one of you helper T cells can help me with this? Once we get you activated, you can start releasing your cytokines, your cheerleading cells, and helping with our overall response. But it can also show off an MHC1 molecule with that same foreign antigen to like a cytotoxic T cell to help activate it. Now, that's where it's a little bit more complicated. But for the most part, when we're talking about cytotoxic T cells, we're saying those are the ones that are inspecting normal healthy cells and looking to see if they are infected from something within. And that's how then they're able to recognize it and generate an immune response and things like that, both to kill it off and then produce a larger scale response. Yeah. Okay, so this slide just has to do with, as I mentioned, the fact that our HIV is affecting and targeting our helper T cells. And the reason I have it bold in there is I think it's somewhere. Is it, I don't know. It's somewhere in there. There's a possible question about that, I think. So it's just an interesting link because everybody has heard of AIDS and HIV and things like that. And it's like, why is that such a problem in terms of affecting people and people dying from it and things like that? Because of its effect on helper T cells. Okay. So as it destroys those helper T cells, then it's going to depress immunity in various ways. Just a few things as we were kind of finishing up as well. Autoimmune diseases. So the whole reason that this immune system works is that we're able to identify self from foreign, right? Is that we're protecting our own and we're attacking things that shouldn't be there. But there are cases, we always are discussing these different systems about, okay, this is how the perfect system works. This is how a perfect heart pumps blood, and this is how coming up the lungs will ventilate and things like that. The immune system should be a perfect system where it can identify every single thing that you're infected with and be able to get rid of it. But we know that's not the case, right? So sometimes you have to take antibiotics. Not for viruses, by the way. Antibiotics are for things like from bacteria. So if you have something, like we said, like a rhinovirus or something like that, antibiotics are not going to do you any good because they don't have an effect. Those are very specific, much like our specific immune system. They can only help with one particular type of pathogen. But regardless, the point is that sometimes our bodies need help. It can't overcome everything that we're infected with. And in some cases, our immune system loses the ability to discern or tell the difference between self and foreign. And it may start attacking our own cells. And that is what an autoimmune disease is where it starts attacking the particular cells of different types of tissues. So you can see in this bottom here different examples of autoimmune diseases. And in fact, if you know about any of these, you'll notice that these are for different tissues, different organs, right? And they're very, very specific. Arthritis, what is that? What does that affect? Your joints, right? Arthro means joint, itis means swelling, right? So arthritis is your joints. This is an autoimmune disease. That's a particular type of arthritis where your immune system is attacking cells of your joints. And that's why those joints are hurting and swollen and things like that, right? So there are different types of arthritis, but that one's an autoimmune disease. Glomerulonephritis, again, there's itis, there's swelling, right? Glomeruli are in the kidneys. Nephrons are in the kidneys. These are specific to the kidneys. So if the kidneys aren't doing their job, they can't regulate blood content and blood volume and things like that. So that can be a problem. Some of these are muscular diseases. Some of these are endocrine diseases. Some of them, diabetes, what does that have to do with? It has to do with the pancreas. And the pancreatic function in terms of its ability to release a certain hormone, insulin. And if those cells are attacked, now you're not producing insulin, you can't regulate blood sugar, but that's an autoimmune disease. Now again, there's different types of diabetes, right? You heard type 1 and type 2 diabetes. Type 1 is an autoimmune disease. So, there are cases where our immune system kind of goes off the rails a little bit. So, whether it's attacking our own system, whether it becomes hypersensitive, like we said, we do have our regulator T cells that can help with that. But we also have things like allergies, right? An allergy, like we were talking about, with allergens versus pathogens, what we have are things from the environment that your immune system is freaking out about, even though it shouldn't be a problem, right? You shouldn't have to worry about like smelling citrus flowers, which may be coming out in certain trees now. You should be able to smell them without it making your eyes itch and your nose run and stuff like that, because those are immune responses, right? Inflammation, extra mucus response, generation and things like that. Histamine being released and causing more inflammation. So those are cases where, again, we're kind of going off the rail. We are increasing our response unnecessarily. And so some cases, you may have more of a generalized hypersensitivity. In other cases, it may be more acute. So for instance, when we talk about like anaphylaxis and anaphylactic shock and things like that, your first couple of times may be like lower level, like you get some inflammation, you get like lower level itching or something like that. But with subsequent exposure to it, again, remember your immune system has memory, so it gets more sensitized to it, and so the response gets bigger as well. And so that can lead to, as we were talking about like in the cardiovascular system, it can lead to even anaphylactic shock. Remember that inflammation is dilating a blood vessel, making it leakier so we can get more of our defenses to wherever we're being attacked. In a hypersensitive reaction, where it's an extreme case, like anaphylactic shock, we're just basically like, I don't know where this thing is, just open up everything, dilate everything. And what's going to happen, right? When we open up all those blood vessels, our blood pressure is going to drop through the floor, because the diameter, right, is a major controller of resistance. And so now with things open, our resistance drops down, our blood pressure drops down, and you may not get adequate pressure to move the blood around the body to get it to all the different organs, right? So in those cases, one of the quick treatments then is what? Epinephrine, right? So epinephrine is then going to work as a vasoconstrictor, so it's going to counteract that effect. It's going to increase heart rate and stroke volume and things like that, right? So that's what we're seeing here, is just kind of tying this system and some of its responses to maybe some of the other systems that we had learned before. Okay. So that is immune system. As I said, when we went lymphatic and immune is a bit more complicated. A lot of it because it has to do with cells and molecules. Rather than focusing on these big obvious organs like we do with the cardiovascular system or something like that, it's all down at cells and molecules. But if we can keep an idea of that organization, right? First line defense, second line defense, third line defense, have some examples of different cells, different molecules in each, and how they help to defend the body, then that's, I think, going to be helpful. First line defense is the easiest one, right? The external defense is second line defense. Okay, now we're inside, so we're responding to certain types of pathogens, but not in a specific way. So we've got things like interferon and fever, and as well inflammation and things like that, different processes, as well as natural killer cells and generic phagocytes that are just eating up foreign things. So that's a little bit more complicated. Third line of defense we saw is a bit more detailed, right? We had that separated in two separate lectures. One focused on B cells and their production of antibodies and how they work, and then T cells and how they do our cell-to-cell combat, right? Our cell-mediated immune system as well. And third line defense, another aspect of it, besides it being very specific, is that it generates our memory, our long-term effect. It builds through time, which is the other reason that's called adaptive, right? It changes, it learns through time, and it protects you more and more and more. Okay? Questions on that? Okay, so that's a new set. So if you didn't review B cells before, you want to do that. Now you need to review B and T cells. You got to be getting those examples down, understand how they work, describing B and T cell function and how it works. Common short answer question possibilities, right? You really want to understand those. Okay? Okay. Well, we will end there today then. Next time, we'll do a little bit of kind of catch up. We're going to move into respiratory system for lecture. And in lab, we move into digestive system as we're finishing up respiratory system today. Okay? All right.